talk about this little lab, and then I can switch back to the chapter review of DNA. Okay. So in this one particular, experiment here, we pour two plates. One we use for three different antibiotic disc to see the diffusion of those antibiotics and how it can kill, bacteria differently. Then the second plate, it's a very easy experiment. What we do, we wash our hands. So we check the effectiveness of washing hands. As you can see, in the beginning, they just talk about normal flora. Normal flora is good bacteria in our body. So you guys can read, for example, our, skin, our mouth, our large intestines, our nose. We have all these bacteria and they keep us healthy. They also stimulate our immune system. But they're just saying that if we have these bacteria in the urinary tract, we can have, UTIs or infections. So we do have some part of our body where we have colonization of these bacteria, but some part of our body like trachea in lungs, we don't have this flora. So good flora, good microbiota, these are the bacteria. They are good for us because they also stimulate our immune system. They cover the surface of our body so that outsiders, any potential pathogens, they cannot come. Even if they come, they don't get the place to live. They don't get the food to eat and multiply. So that's the purpose of good bacteria. You guys can read a little bit about it. And chapter 16 and 19, we will explore more on those chapters about good flora, and host microbe interactions. A transient, so you can see here resident and transient flora. Resident flora is the flora which is always living on our body. However, transient means it comes to our body, but it's not about good or bad flora, but they have found that sometimes good bacteria are also present on our body. Transiently means they come and go. So that's the meaning of transient flora. And what they want us to do in this experiment, it's hand washing. So you can see you have this agar plate. You can label it on one side unwashed and the other side washed, or you can say before soap and after soap, means after washing with soap. You can use your thumb, you can use any finger. It's not up to you, you have to follow the same. If you want to stick to what they say, it's fine, but you guys can use any finger of your hand before and after. Make sure that here they say, oh, completely dry your hands with paper towels. But let me tell you that in my face-to-face classes, what we do, we take a Petri plate and we just label one side unwashed and the other side washed. And before, on unwashed side, before washing our hands, I just stick one of my finger on one side, press it on one side of the agar plate, just gently for maybe 1 minute. And after washing my hands with soap for 20 seconds, I come back and let it dry completely, air dry. I don't touch anything because that may induce or include or contaminate your fingers. Although water doesn't have any bacteria in it, but you never know, just to be careful. After air drying your thumb or your index finger, just press on the post side, means after washing your hands with soap. So I want you guys to ask, what do you expect before and after washing the hands? Because now you have an idea that that's how bacteria grow, and what should you expect? What do you think what should happen? Will you see growth after washing your hands on the Petri plates? What do you think? If you understood this experiment, that you're testing the effectiveness of washing hands. So what do you think? Like, after you wash your hands, put your fingers on the Petri plate, will you see any colonies on it after washing hands or no?
No.
All right. Okay, anybody else, guys? What do you think? Hmm?
I think no.
No? All right. Okay, so I'm going to show you, this is my face-to-face class that we are doing experiments. So this one is ubiquity of microorganisms. So what we took is we took the plates and we divide them into four different quadrants, and we use the swab to take the samples from any surface, like 1, 2, 3, 4. They might have explained that one is shoe. As you can see here, this place here has a weird, looking this colony, but this is a kind of bacteria, or it may be a fungus. This is a yellow colony, yellow colony. This one looks similar. By by looking at these bacteria, I can tell that they look similar because the type of colony, visually I see they are the same. The size is also same, means they are dividing at the same time. But yellow and these are two different. This one is another one. Then some of them are coming very slow. Similarly, on the other plate, it's the same plate, but you can see from the back side or the front side, there is some moisture here. So you can compare the morphology of these cultures, and you can find out that these are different bacterial species. So these are, and they have to answer all the questions. So they are doing really, you guys are also doing awesome job. So here is the plate where two people were sharing this plate, and we divided into two quadrants, sorry, four segments. One is unwashed. This is the wash, and the third person is taking unwashed finger and washed finger, or it may be vice versa. So I want to tell you that if you look at the top one, and you are expecting that after washing hands, I should not see bacteria, but we still see bacteria. Do you guys know why we are seeing bacteria on our hand even after washing the hands? What do you think? What happened? That soap didn't work, or what could be the
Maybe
reason?
certain areas,
Mm-hmm.
or like you just didn't wash your hands properly.
Okay. But it's really amazing, right? It's like strange,
Yeah, it just
right?
shows you like how bacteria can be that strong, and even if you wash your hands, it's still gonna be there.
Mm-hmm. Anybody else, guys? What do you think about this experiment? So this is the same plate, but in the front. But one thing you can see, this colony here, this is the unwashed, right? Can you see? There was something on the hand or that finger, and after washing, it's gone. It's not anymore here. These bacteria, like they look like similar, like, present on the hand. These species look same on both sides, right? Whether you wash hands or unwashed hand. Can you see? Because if you look at their morphology, they look the same.
Isn't there like also like good bacteria or like bacteria that is normal?
Mm-hmm. Okay. Yeah, I'm coming there. That I just want you guys to critically think about it, because see, unwashed, you can see that there are a lot of these bacteria, but now yellow ones are more. They look more here, right? But some of them are maybe washed off. So what happens is, like, purpose of hand washing is to remove any extra dust particles, anything that is on your hands or fingers, right? 20 seconds is very, very effective in washing the hands to get rid of anything that is external. But what happens is that when your fingers are wet and you have in your groups your bacteria, these bacteria are before hand washing and they still stay there, right? Only thing that was going off, and some other bacteria, maybe different species, because they don't belong to our skin. But our skin is rich in all good bacteria. So on both sides, you have their resident bacteria. These are external something on your hand, which is washed off with soap. And that is the purpose of hand washing. So you will never see completely washing or like all bacteria are washed off from, because our fingers, those little groups also have a lot of bacteria in it. That makes sense? So you guys will be able to answer those questions. I have another one from my other student. 1 second. I think this is the same report. Okay, they are sharing the plate, so maybe they are the same, okay? Here also you can see a lot of bacteria washing and hand washing, because when you come with the wet hand, you see more bacteria here as compared to when it was dry, because that wetness also leads to all the yellow ones are coming more here, the creamish ones are more. But the other bacteria who are not permanent resident on our body, they will be washed off. So that's how it's one of the universal effective technique to wash off the hands. All right, so did you guys get a chance to look at this chapter, DNA, structure and function? Or no, you were preparing for the exam? Hmm?
Didn't get
So
that, Professor. Was that a question?
I'm asking, like, did you get a chance to look at this chapter or not?
Oh, yes, but not. I'm still
Um,
going through it before the Wednesday.
Oh, okay. Yeah, so the recording is there, and I will explain in, let me make it a little bit simpler for you guys. So when you come back to this chapter, this is the chapter that you will need to spend more time, because, sometimes the DNA people really haven't touched that unit in life, maybe, or they did it long back ago. So it will be a first time, okay, refresh it simple, then go into the details. I will not be asking you chemical structures, because that's what we do in biochemistry. So all those structures will be there, like you can see this slide, but I will walk you through, like, okay, what is needed to be done. Before I start this chapter, I had one question. So keep on doing the labs. I did post two extra credit opportunities for you guys. So I would say that everybody should do it. One is case study, which is based off your chapter 5 and 20. And there is another one, a discussion post. So let me know if you can answer the discussion post, and just make a summary of what you learned so far in those chapters which I mentioned in the discussion post, and you can reply to two other peers, and it will be extra credit at the end, like means I can add those for so those 5 points. And case study is 10 points, but it's a good case study that you will be able to answer easily, that how we use antibiotics, and sometimes we have resistant strains, antibiotics don't work. So, like MRSA. So understand the case study, and, answer the questions which are following the case study. So these are two extra credit that you should be working on by the due dates. Please check the dates, because right now I think you have enough time, but check so that you can submit it on time.
Professor, the discussion one, I can't see it on Brightspace.
Okay, let me check. Well, give me a second, okay? All right, so chapter 7 and 8 are mostly like 8 is a continuation of chapter 7. So let me see where I can start. So, this chapter starts with some, history, that so you guys can, look at that part yourself. I will start with a couple of terms here, like for example, chromosome, genome. And when you look at the genome is sum total of the genetic information of any cell. And what does that mean? Means it's sum total of the DNA inside any bacterial or any eukaryotic cell. And it's like inside the cell, you're looking for the presence of DNA, whether it's present on mitochondria, whether it's on a chloroplast in eukaryotic cell. And we have DNA as chromosomes in the eukaryotic cells. Genome of viruses, you can see it can be DNA or RNA because viruses don't have nucleus, they don't have any other organelles. But when it comes to these three structures compared in one slide, you can see this looks like a plant cell or cell that has chloroplast. They also have mitochondria, they have nucleus. So the genome of this eukaryotic cell will be sum total, mean this plus you add the mitochondrial, we call it mitochondrial DNA, DNA, then you can say CH chloroplast DNA. So this sum total will be the genome, means these genes plus chromosomal genes, those are the genomics of this particular cell. And there is additional, sometimes you see these small extra chromosomal DNA that in plants also, in eukaryotes also we found, we call it as a plasmid DNA. Plasmid DNA is not needed by these organisms, but they may have some extra genes. They may give them some advantage in some other, unfavorable conditions. Like bacteria mostly have plasmid DNA, and it's good to have this plasmid DNA with some extra genes. Like here you can see most of the genetic engineering work is done by this plasmid DNA. And my day also started by isolating the plasmid DNA, or we had the ready-made artificial plasmid DNAs. You cut this plasmid DNA open and you can insert your gene of interest and you can study, you can put them back into a bacterial cell. And bacterial cell, when it multiplies, you can replicate your DNA, just making the copy. So you use bacteria as a medium or media to just, propagate or make copies and copies of your gene of interest. Here they're just trying to show you a virus. It's a DNA virus, and this one is an RNA virus. That's their genome. What are genes? So your DNA, it's a double-stranded, let's say some length of the DNA is there. The pieces of DNA, like there is a smallest unit of heredity, we call it as a gene. So it's the smallest means it's complete in itself. And when you express this gene, it makes the messenger RNA. And when this messenger RNA is expressed further by translation process, you make a protein. So we call this as a gene. Gene means it's capable of making a product that can be a protein, it can be RNA, both, but it's a smallest unit of heredity, or you can say fundamental unit that can be, transferred from parents to the next generation. The type of genes we have, like if you look at the 4,000 genes in bacteria, or you are looking at the 20,000 or 30,000 bacteria in humans, some genes, they are known as structural genes because mostly they make proteins, they code only for proteins. Some genes, they code only for RNA. For example, so structural genes, they make messenger RNA, and messenger RNA further makes the protein. So always they are responsible for making proteins. There are some genes who makes only ribosomal RNA, let's say ribosomal RNA, and like ribosomal RNA is responsible for making ribosomes, but they never make protein. It means this RNA will never be translated into protein. And we have transfer RNA, and transfer RNA has the role in bringing amino acids, or we say transfer of RNA during translation process. So these are the genes, they code only for RNA. They never translate further those RNAs into a protein. And there are some genes who are responsible for controlling some other genes. So we call them as a regulatory genes. For example, you have a gene here, gene 1, and you have gene 2. But there is this gene here, we call it regulatory gene. And this regulatory gene is making a protein here, let's say this protein. So this protein can go and control other genes. So that's why the name is regulatory genes. What genotype is? Sum total of the DNA, means all the genes in an organism. We call it genotype. When you express, this genotype, what do you see? A phenotype, external, like you can see these traits, you can observe them, but it is as a result of expression of the genotype. The genes give you the phenotype. What is central dogma of life? It says that information goes from, because in the beginning, when we didn't know about, like, okay, DNA is there, DNA carries the genes. At that time, many different labs were asking this question. At the very end, they found some correlation that if you have genes, if you have DNA always makes a copy of itself into RNA, and this process is known as transcription. And when you translate, so you can see it's a double-stranded here, then you make something like single-stranded, which is mRNA, messenger RNA. It's like a message. And from messenger RNA, you make this protein. So there are 2 steps in gene expression and flow of information. We call it central dogma of life, that it goes always from DNA to RNA to proteins. There are some exceptions. We call it as a reverse genetics. Means it will never go from DNA to RNA to protein. It says RNA to DNA, then DNA to RNA, and then it makes the protein. So this is a little bit reverse genetics, and mostly example is viruses, and HIV virus is also an example of. We also call this as a RT virus. At some point, we did discuss in chapter 20 that HIV is a RNA virus. And when it integrates its DNA, so they have this enzyme reverse transcriptase that can actually make DNA from RNA, right? And that's why we call it as a reverse genetic. So central dogma of life is again shown here, that you have double-stranded DNA. This DNA is opening up. This is the process where they are making mRNA. mRNA comes, it's a eukaryotic cell, so it's coming off the nuclear pores into the cytoplasm. And cytoplasm, you can see they have tRNA, they have mRNA here, the yellow molecule. These green ones are the amino acids. So tRNA is charged, as you can see, this tRNA, they will be charged with these amino acids. And here you can see the ribosome sits on the ribosome, on mRNA. It moved from here, the start point, and going forward, reading the message which is written in these nucleotides. When they read this message, what they are doing, what is coming off, this is a chain of amino acids. So they just make the bond between amino acids, and this is a protein. And protein just fall apart like at the end when this translation process is complete, they are making the protein. And this is another version, like where you can see ribosome is sitting on the mRNA, and tRNA, this molecule is shown as tRNA. And these are the amino acid. They are making a bond to one another. We will talk about this process in a minute. So just wait until then. But this is a central dogma of life, that information goes from DNA to mRNA to proteins. Now, in this chapter, actually, there are 3 processes which we need to understand. One is replication. Replication is needed only when the cells divide. Gene expression is when, the first step is gene expression is transcription. So information goes from DNA to RNA and then RNA to protein. So these are two steps of gene expression. And the first part, DNA self-replication, is just prior to cell division. So a little bit about the structure of DNA. DNA, like it's double-stranded. I will explain this slide to you. So this is the structure of a DNA. What DNA is made up of? Nucleotide. So what is a nucleotide? Nucleotide is made up of these. This is one nucleotide. They have a nitrogen base NB attached to number 1 carbon here. Number 1, number 2, number 3, number 4, and this is number 5. So this is there are 4 letters: thymine, guanine, cytosine, and adenine. If you have A, for example, this is T here, thymine. This is the structure of T. Okay, it's like a hexagon, one closed ring. Attaches to number 1 carbon here. This is the sugar. If it's a DNA sugar, there is one more difference between DNA and RNA. DNA is known as deoxyribose. So I want to tell you that what does that mean? If carbon has OH group here, it's a ribose sugar. If this oxygen is missing from here, if this oxygen is missing, only H is present, we call it deoxy. And deoxyribose is present only in DNA. That is the one difference. DNA is double-stranded. You can see there are 2 strands. RNA is single-stranded. Okay, so we'll take off this. So these are the 2 strands. It's like a ladder, but if you twist this ladder, it will become just like this double-stranded DNA. That's how we, draw it. And in the middle part, we have these nitrogen bases. T always pairs with A. T is complementary to A by 2 hydrogen bonds. These dotted lines are nothing but hydrogen bonds. G always pairs with C by 3 hydrogen bonds. So you can see 3 hydrogen bonds or 2 hydrogen bonds, right? And these hydrogen bonds are keeping these 2 strands together. So just going slow a little bit. So what you can see this part here. So these are that's where I started, that DNA is made up of nucleotides. So this is one nucleotide, a nitrogen base attached to this carbon, and there's a phosphate group PO4 at 5' end. So what is 5' end? When you name these sugars like carbons, 1, 2, 3, 4 at this corner, and this is number 5 carbon, and we say 5'. 3' means what is on 3' end. So this is one nucleotide, and the other nucleotide comes, this one, attaches itself on 3' end here. That's where they add. The other one, it's like your left and right hand. Left hand, right is this one, left hand, and this is my right hand here, this part. So who is going to make this chain? Actually, people are coming, these nitrogen bases, and they attach themselves at the 3' end. Then another one will come here, then another one. So always there is a growth of this chain always in 5' to 3' end. And when you make the other strand, it runs in opposite direction, as you can see. So it's like you are making a ladder, but now the second part of the ladder, which is this strain, it goes from 5' to 3'. So one runs in 5' to 3', and this runs 3' to 5'. And we call this situation as an anti-parallel. So DNA is, we say, it's double-stranded, helical, and anti-parallel. These are the 3 things that describe a DNA. It's a double-stranded, anti-parallel to one another. It's complementary because A always pairs with T, G always pairs with C. And we also call them these single rings. As you can see, thymine. here and cytosine. These are single rings. We call them pyrimidines. Even USL is also pyrimidine. And we have two other, like adenine and guanine, like these green ones here. We have two rings here, 1 and 2, G and C. So we call them as a purines, because in chemistry we have these structures. That's what they match for. Purine means adenine and guanine. They match this structure with double rings, number 1, number 2. And if you look at pyrimidines, you look at number 1, only one ring here. So look at thymine. Thymine looks like this picture. Cytosine looks like this picture here, or this image, or this structure. So we call it as a pyrimidines and purines. So always remember nitrogen bases. Nucleotide is when nitrogen base is attached to the sugar and to the phosphate. Those three parts of the nucleotide, and you keep on repeating these nucleotides here, nucleotide, you call it as a nucleotide chain or one strand of DNA. And on this side, you have the nitrogen bases hanging, and the other strand comes, and both of them come together and put together by the hydrogen bonds. Here is just a picture to show you that there are two hydrogen bonds here between adenine and thymine. There are 3 hydrogen bonds here between guanine and cytosine. I won't be asking you the chemical structures here, but if you need to go there, you guys are always free to learn more, but I will not be asking those questions. This is just to show you that how one nucleotide, because here is here, the other one is coming at 3' end, then 5' to 3'. So that's how we make, the nucleotide chain. It always goes from growth of nucleotide chain means addition is 5' to 3' end, never 3' to 5'. You cannot add anything on the 3' end. Just remember, this is the reactive group, one free OH group. That's where you can add something and make a ester bond. So sometimes, we come across this phosphodiester bond. Now, ester bond is a bond between hydroxyl group, which is present here, OH, and this was OH actually of the phosphate group. And like there is a condensation going on, so water will be released, and we make this bond, we call it as a ester bond. But if you need to go there, only then it's required. Otherwise, nobody's going to ask you that. So this is the structure here, anti-palo, you guys can do that. Now they're talking about the RNA, because if you understand the structure of DNA, RNA is easier, because RNA is nothing but it's a single-stranded. That's it. This is single-stranded. You have nitrogen bases, let's say A, U, G, and C. Now, U is uracil, which is not present in DNA. So this is the third difference, that in place of T, T is in DNA. And in place of D, we replace it with U in RNA. U stands for uracil. So this slide here talks about the differences between RNA and DNA. Here, DNA is double-stranded. Here, it's a single-stranded. You have uracil. Here, you have thymine, right? If you look at the sugar here, ribose sugar, it's D. D means oxygen is missing. It's highlighted here. Look at that. It's a normal sugar, but this oxygen is missing. So we call it as a deoxy, and it's present here as a hydroxyl group. So those are the differences between DNA and RNA. Now the next part is about the DNA replication. So I can quickly tell you that they are talking about the replication because, this cell in bacteria wants to divide into 2 cells. So the first thing they have to do is replicate their chromosome. So replication of chromosome means they need to replicate their DNA. But how will you start DNA replication? Is there any place? So there is this specific place, we call it's highlighted in this part here. We call it origin of replication. Sometimes we call it ORI. And DNA opens up. For example, let me give you, this is a circle, right? There will be another stage here that this circle opens up like this, and this is double-stranded. So one one enzyme will go on this direction, one enzyme go in this direction. It's like a field you are running. You come here and you cover all that field, means lay out the second copy, and you meet. It will be faster. So what you can see, we call it as a replication fork, and you keep on opening this helix and you start making the lighter. So you can see you start making the, first you separated these strands and you start making the second strand against the older strand, right? And you do the same thing for the other one. So in the end, you will have something like these two circles, one old, one half. So in these circles, what you see, you have old DNA, old strand, and this one is a new strand. So we call this as a semi-conservative, because in two daughter cells, one is original strand and one is new, old and new, old and new. That's how DNA replication takes place. But there is a little bit more in here. It's not only this much. This is one more time they're just trying to show you that you open it up. It's like you have a helix and you're trying to open it up and starting to lay down this new strands. We also call it bubble or eye. I wanted to tell you that there are multiple ORIs in eukaryotes. It's not only one, because an eukaryotic DNA is linear. It's like, a lot of, so you will have one bubble here, one bubble here, one bubble here, and they keep on, multiple enzymes are carrying out the DNA replication. So it's feasible to do, 6-foot long DNA replication in the eukaryotic cells, for example, human cells. Here is another slide where you can see two strands. This is your original, the blue colors are shown. These are two strands of DNA. And you separate them. For example, you separate them and you start making T here. In front of this T, you start making A. So T here, A, right? So it's shown daughter strand and one of the daughters, double-stranded DNA made from the old strand. So one is kept as blue, old. This is new and old, old and new strands. And if you look at the information, it is the same, AT, AT, GC, GC. There is no, so you're just copying the DNA, but we call it as a semi-conservative DNA replication. These are the enzymes you guys have to know their function. So let me tell you that these slides, they talk about the DNA replication, but I'm going to explain all these slides myself by using the, my drawings. So you have DNA, which is double-stranded, and let's say origin of replication is there. This is the part of the DNA, right? Double-stranded. And this is your origin of replication, let's say. That's where the replication will start. It's a starting point. I have opened up this DNA, and there is this enzyme, we call it. This replication will go in two directions. We call it bidirectional. Because, one enzyme will do it in this direction, the other one in this direction. For simplicity, what I'm going to do is I'm going to divide it into half and show you that on one side how the replication process starts. Because it's very complicated process, you will be lost. So go slow, 5' to 3' end, and 5' to 3'. These are two strands. Now this enzyme actually is opening this helix because it's opening, it's like unzipping. So this enzyme is known as helicase. It unzips the DNA. So it's going in this direction, right? Now this enzyme I will just get rid of these drawings because I need space. So there is this enzyme, we call it DNA polymerase 3, Pol3. This is the major enzyme which will do the synthesis. So these are old strands. This is also old strand. Now you're trying to make a new strand, right? This enzyme says that I will work only in 5' to 3' direction. And they say that I will not start myself, not initiate. They need a starting point. So I'm using this color here. So I'm saying that this is your old strand. In front of that, the direction will be 3' to 5'. And this enzyme is saying that I will work only in 5' to 3'. So in the bottom strand here, 5' to 3' direction is okay for this enzyme. They can start. So what else do they need that I cannot start? I cannot add, let's say you have A here. Ideally, it should add T here in front of it, but it doesn't do that. So there is one more complication here that they need a starting or starter. We call it as a primer. So this enzyme here, primase, makes this small molecule, RNA molecule. We call it as a primer. Now, primer means RNA and RNA means uracil will be there. So if you have A here and let's say T, G, C, T, this is your DNA. So this enzyme primase will go and start making A to U, T to A, G to C, C to G, and T to A. This is your primer. So primer is synthesized. Now DNA Pol3 DNA Pol3 is okay. It comes and start adding new nucleotides here. Continuously. As it's opening, it goes and start making this. So synthesis of the bottom strand, because it's 5' to 3', we call it as a leading strand. We also call it continuous. Okay, because it's happening continuously. Means once you started, primer means it's like you want to put the primer on the walls, then you paint them. But now we have everything mixed up and you can start adding the paint to the walls. But usually in earlier times, we had the primer. We had to add the primer so that you can stick to it. Same thing is happening here. You're starting the primer first, then the DNA synthesis. Now, let me get rid of a couple of things here so that I can show you the top strand. Because top strand, the complication is that it's 3' and I cannot do 3' to 5'. I cannot start it. So what is the option I have? The same very enzyme has two hands. One hand, they are doing this process, one side. With the other arm, they just switch it to, a process. We call it as a, let me see. So because this is opening in this direction, right? As it's opening and direction is different. So 5' end is here and 3' end is here. So they start with the primer and I will use, so one primer and they opened it up. Then another primer and they opened it. Like, that's how they start. They need multiple primers to do the synthesis. So this is known as discontinuous. They also call it lagging. They also call it Okazaki fragments. Are you guys able to follow me or no?
Yes.
Okay. So
We're following.
yeah, because it's discontinuous. You will have to have your eyes there when I'm drawing it and listening, okay? Discontinuously lagging and Okazaki. These are the three names for the top strand. The reason I told you, because see, it's 3' end here. So definitely 3, it can't go this direction. So it is done in this direction, but it's a slow, or we call it lagging strand. Although in the end everything matches at same point, means this will reach at the same time as compared to the leading strand. But you have to keep in mind there are a lot of factors here. You're opening your strand this direction, so you are not going in this direction, means you're not going this direction. You're opening DNA on this direction, right? There is another enzyme on this side working, so I took only the half of it. But it is not still finished. We need two more enzymes because see, our DNA has uracil in it. So we have to remove these. So there is an enzyme, we call it DNA Pol 1, Pol 1. And this was Pol 3. This is the major enzyme which does the major addition of nucleotides. But DNA Pol 1 removes the primer. These are two things it does. Remove primer and add DNA nucleotides. So it will remove these primers and it will add these nucleotides. But there is a gap between the two bricks here. Like for example, if you're removing the primer and adding, let's say you removed and you're starting adding. So there will be a gap between this one and this one, like this one and this one. So to fill this gap, we need another enzyme, ligase. I will show you a video in the end, like maybe at that time. So ligation is nothing. It's like you're taking, you have two bricks here and let's say three bricks. You remove this one away and now you're starting to, add something from this side. So you keep on adding, but here you stop. And there is a gap between this brick and this brick here. So to fill this means attached to one another, there is another person who is coming and doing the job. And we call this as a ligase enzyme. So once you remove these primers, the blue ones, and you start adding, everything will be just like the DNA. In the end, because you want to remove the primers and add the DNA nucleotides, two more enzymes come into play, DNA polymerase 1 and ligase. So that completes your DNA replication. Know each enzyme and their function for your next exam. The next part of the chapter talks about gene expression. Gene expression has two steps, transcription and translation. So out of two, so I will write a sequence of DNA for you guys. And if you guys want to practice with me, it will help you to understand the transcript, your extra credit as well. So 5' to 3' and 3'. So you can randomly write any sequence. I will say A, T, okay. So I wrote only 3, but you guys can write any length of it. Can you guys write the complementary sequence for the top one? So write the second strand of DNA. From this DNA, we are going to make a protein, okay? And we will also understand the naming of a couple of things we do. Can you guys do that on your end?
Yeah.
So this is our DNA, right? This is our gene. So what we are going to do is from this gene, genes, what they do, they use only one of the strand. It can be this one or the bottom one, but never both. So there is this part here, we call it as a promoter, although this is also promoter, and there is a terminator site. On any gene, terminator. It's DNA, but this is promoter. Promoter is where this enzyme RNA polymerase will bind. I will say polymerase only. RNA polymerase means polymerization of RNA, making RNA from DNA. Termination means that's where it will stop, right? Because you have another gene here, gene 2, and you don't want to transcribe it. Right now, you're making a protein out of this gene. So this strand here, so you guys can fill these gaps. I have C2G, T2A, G2C, A2T, T2A. This is complementary strand. I'm going to use the bottom strand. I call this strand as a template because my template here is this strand. And what I'm going to synthesize, 5' direction is always, whether you're making DNA or RNA, it is going to be always 5' to 3'. So let me use a different color here for simplicity. And I'm making RNA, 5' to 3' with the help of this enzyme. So this will also open these two strands, and only this against this one, you start making this complex to T2A, A2 uracil. That's very important. So what I start writing from template, you start making mRNA. mRNA is nothing opposite of your template strand. So T2A, A2 uracil, C2G, G2C. Again, what should I write here? Can you guys tell me? What should I write here? On question mark?
T or U?
Uracil, right? And here also uracil. Whenever it's T, you have to replace with the uracil. Okay, so now if you have this, now scientists, when they looked at the transcription process, if you look at the picture, that's easy, you guys can go in detail, but I will show you how to make a protein out of it. So scientists found that cells keep these nucleotides. If you have let's say your gene is 1200, or 1500 nucleotides long, nitrogen bases are there. They found that your amino acids are not 1200. They don't code 1200 amino acids. They code something like less than that. And you have only 4 letters, A, G, C, and T or U. But you have to code 20 amino acids naturally to make a protein, right? So to understand that, let me tell you that what cells do, they keep this as these nucleotides on mRNA, they make a code. This is known as a codon. The combination of 3 letters, it codes for one single amino acid, which is methionine. Then they use this code, like these 3, code second amino acid, then triplets, codon. And they keep on joining one amino acid to another one, amino acid to 3, amino acid 4, and that's the protein. This is the peptide bond between amino acids, which is made by actually ribosome. So for translation process, actually, first thing is to make the mRNA. You need mRNA. You need amino acids, so tRNA. I'm making tRNA, like this is amino acid put on the head of tRNA. And there is this part where they match with these codons. So tRNA comes and match. They say that, oh, I got the right amino acid. They deliver it. There is a ribosome here, small and large subunit. Ribosome will move, read these codons. tRNAs come here, and ribosome makes although this is big enough, actually, they join the amino acids. And this empty tRNAs will go out of the ribosome, and that's how translation starts, and it reaches at the end where you have stop codons to stop the process. So let me put that in a minute together. I know it's not that easy, but I wanted to know for you guys that you have to understand that this is the mRNA transcription process. Then cells use them as a codons. Now I will go back to the chapter. After transcription, here you can see the transcription. This is a gene. You have promoter, you have terminator, and this is the process. I think it went somewhere here. Yeah, here. So ribosome, not ribosome, RNA polymerase. This is an enzyme. And there is this small protein, we call it sigma factor. It attaches, make sure that it's bound on the right promoter, excuse me. And it opens up a little bit. We call it open complex like this one. You don't need helicase here because this enzyme is different. It reads, as you can see, the DNA comes back to normal position. They just open it up and use the bottom strand just to read the bottom strand and make the transcript. So yellow molecule here, not yellow, actually pink. As you can see, they are highlighting that C to G, T to A to U, which we did already. And DNA remains back to normal position and mRNA has been synthesized. RNA polymerase is recycled. This is your transcription process. Now they are saying that in transcription, for some genes, the direction of promoter is in this direction, the other one is in this direction, right? So for this gene, they are using the top strand as a template, but for this gene, they are using the bottom strand because, direction is very important. 5' to 3', then this will be 5' to 3'. And similarly for this gene. So you have DNA, right? You have long DNA. This is one gene. So top and bottom strands are there. For some genes, the top strand is acting as a template, or for other genes, the bottom strand is acting as a template. So when you read these slides, I want you guys to go slow and understand what I'm talking about. So here is a translation process shown here that this is your mRNA. And on mRNA, the ribosome is an enzyme. I want to tell you that here no other enzyme like DNA polymerase or RNA polymerase will come, but ribosomal RNA act as an enzyme. We also call it as ribozyme because it's an enzyme. It's a fancy name for the ribosome. And this is the mRNA. They have a region, actual gene. But before that, we have a couple of, sequences here like start codon, this sequence here, which will interact with the ribosomal RNA so that ribosome knows that that's where I have to start. And there is a stop codon. I will talk about that in a minute. And this is the protein. These are the three names, three letters: serine, histidine, cysteine, tyrosine, serine, proline, glutamine, serine, methionine, leucine. They attach and make this like a necklace. Reading frames possible. So here they say that reading frame of the protein is very, very important. I think cells know what is the correct reading frame because if it's here and there one nucleotide, you can see they are making three different proteins. But cells know that which one is the right frame for the protein because even if, because ribosome is reading 3 at a time, the codon. But if you read it differently, like UGG rather than CUG, you're making coding different amino acids here in a protein. This is a very important part of your translation process. We call it as a genetic code table. So you have 4 letters, A, C, G, and U. Cells keep codon as 3 letter as a code for coding amino acids. So if you start making every possible combination, but you have to choose 3 nucleotides or 3 letters, right? So let's start making these. I will say A, C, G. I will start using them and make every possible combination. I will say O, A, and you can repeat them also, okay? G, 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 C, U, G, U, C, G. I will start making them, and scientists, when they exploited all the possibilities, it came out to be 64, which is shown here. 4 plus 4 plus 4 plus 4, 16 fours are 64. So 4 letters, possibility are 3 is equal to 64. Out of 64, these are 3, UGA, UAA, and UAG. They do not code for any amino acid. We call them as a stop codons. So, ribosome stops making any or adding any amino acids to the growing chain. AUG is known as a start because all the protein, they started with methionine. It codes for methionine as a first amino acid. So this is a genetic code which is followed by all the living organisms, including viruses. We call it universal code. But this code is a little bit strange in the sense that we needed only 20 codes, but we have more. And why is it so? I think cells wanted to keep it very, very accurate. They didn't want to have a chance of any mutation. So as you can see, I will say some couple of statements here. This codon codes for amino acid, glycine. This also codes this one, this. But they do not code for anything other than this amino acid. So one codon, one amino acid. But there are some amino acid like glycine. They are coded by more than one amino acid, one codon, sorry. Right? So there are many, many amino acids like glycine, this one, lysine, lysine. They are not coded only by one, but they are coded by many of the codons. But this codon codes for one amino acid. It's not like the GAA is not responsible for making this or that. It's only responsible for making one amino acid at a time. So these are the properties of genetic code. This is the structure of tRNA. I was drawing it like this for simplicity. Like this is the tRNA, the anticodon loop. We call it anticodon because codon is present on mRNA. And they get charged, with the amino acid. So amino acid charging means this is the single RNA molecule which acquired this stage or this conformation. We call it as a hairpin loop-like structure on one end. So it's like RNA folds on itself, but this is the loop structure because they don't have complementary sequences. They make loops like this. And this is known as anticodon loop, which interacts with the codon. So anticodon and codon, they interact with one another. And the purpose of tRNA is to bring the amino acids. Or we also call it transfer RNA. So transfer RNA means it transfers the amino acids to the protein synthesis machinery. So we need mRNA, we need the amino acids which tRNA is bringing. And here you guys can see that translation in prokaryotes begins before transcription is complete. So here you can see this is the DNA of the prokaryotes and RNA being synthesized. But see, they started to make the protein because, it's happening in the cytoplasm, right? Everything is in the cytoplasm hanging like this mRNA, and they start making the protein right away. So we call it as a simultaneous process of transcription and translation in prokaryotes. So you can go over these slides about translation, but I will show you what translation takes place, in this part here. Initiation. This is from your Bio 1-1. I took it from there. I like these better. So this is just a review for you guys. You have already done this chapter. So if you look at this part, the small subunit comes and bind on the ribosome. And this is your P site, of the P site, and this is A site. This is their AUG. And methionine is the first amino acid. This is the anticodon interaction. And then the large subunit comes and sits A to A, P to P, and E exit site is present only on large subunit. So they make this complex, we call it initiation complex. You have the first amino acid at the P site. Now the second tRNA will come here. They pair with the codon, and the second amino acid comes here. The next step is to make the bond between the two. So let's see in the step-by-step process here. So we were here. This is your first amino acid. Just forget about these. You have this tRNA coming, bringing the amino acid. And now, because both of them are together here, now it's time to make the bond here. When you make the bond, you have to transfer this amino acid to this one. And this tRNA is empty. It has to go here and from E exit side. So you can see this one, both of them, they go like they translocate backwards. So this one from here comes to this one, and the E site, they exit. And now in place of this one here, new amino acid means new tRNA will come. And this cycle keeps on going until the stop codon is coming, right? So they keep on adding, and look at this place, like this slide for a couple of minutes. You need to focus on it, what I explained, like how it's happening. And now it's the end, that ribosome reached the end where, all these things are in place, P site, A site, and E site. So A site tRNA is occupied by this protein release factor because there are stop codons UAA, UGA, or UAG. Any of them, if the ribosome encounters it in the A site, it is time to terminate the translation process. So you can see what has happened. The tRNA has come off, the polypeptide is free now, the large subunit is free, small subunit is free, this protein is also free. Everything disassembles. And they can come back. Actually, ribosome is recycling of ribosomes, because they can start again, And this protein will fall and start folding into three-dimensional structure. That's how you can say these are the protein chaperones. They help in folding the protein. The next part is, if you look at this picture, I have multiple same slides actually. Here you can see in prokaryote transcription and translation takes place in the same place, cytoplasm. Transcription takes place in nucleus in eukaryotes, and translation takes place in cytoplasm in eukaryotes. Yeah, just know those questions. All right, so these are the comparison of eukaryotic transcription and translation. So let me go, so this slide and this slide is the same. What you see here, it's found only in eukaryotes. Eukaryotes have, their genes are arranged in exon, introns, exon, intron, exons. What are these? This is a coding part of the DNA. This is non-coding. So their DNA is always long, very, very long, or unnecessarily, like, there are some segments of DNA, they don't code for anything, coding and non-coding regions. And we don't know why it's like that. So in eukaryotes, we have unnecessarily these pieces that I don't need them. So what they need, first thing is that they transcribe as it is, and you do have coding sequences, and in between you have these non-coding that you don't like them. They don't code for anything. So in order to remove them, we use this term splicing. You splice them off. Now what you see in the end, you have this complete. If you join this part to this part and this part to this part, you will get this mature RNA. This is known as premature or pre-mRNA. And there is a little bit one more type of thing we have to do. We add this cap and the tail. Tail is made up of like approximate 200 adenines are added. Tail, or we call it poly A tail, which is added at the end or 3' end of the mRNA. Now the purpose is why? It is to protect this mRNA, because this mRNA in cytoplasm, it has to go out of the nucleus. And nucleus is actually enzymes, they are ready to, eat, or you can say destroy these nucleotides. So cap and tail protects this DNA and also stabilizes this mRNA, actually the message in eukaryotes. So that's the a processing which happens in eukaryotes, not in prokaryotes, because they don't have introns or anything like exons or introns together. Everything is like in the exon form. It's going to code for something. And this is another slide. You can see the yellow regions have been removed and the blue regions have come together as a coding part of the protein. Bacteria don't have any processing tools to do that, and neither they have introns in their DNA. All right, so that's the translation process. All right, so do you guys want a break? Okay, so the next part, we just talk about, because, now we saw how DNA looks like, how it's expressed into proteins, and there are different other factors that you can compare between prokaryotes and eukaryotes. In this slide, they're just trying to show you that how gene expression can be controlled. Like, you can make it more proteins out of that gene. For example, this gene A, you can see that, some protein is being made, like basal level of expression. This gene is kind of off, means it's not making any protein. This gene here is making a lot of mRNA, a lot of proteins, because there is a need for, gene C or gene protein for that expression. It may be a protein that is needed all the time, like cellular respiration and more quantity. So these are the names of certain enzymes or proteins. Like constitutive means constantly being made. So genes are always on. For example, glycolysis, Krebs cycle, because it's going continuously. So those genes are needed. Those proteins are always on. Inducible enzymes, and this is the last part of this chapter that we need to focus a little bit more in detail. So inducible enzymes are not routinely produced, means they are off initially, and you need to switch them on. On the other hand, these enzymes repressible are in on mode, and you have to switch them off. That's what, if you can understand, I think that analogy will go long way to understand these two types of operons. But before that, there was something in here. I'm not sure if it was here. No. All right. There is this term. Where did it go? Okay. There is, I feel, one slide even I'm missing or something. All right. Before I talk about this part, I want to tell you that in eukaryotes, every gene, for example, this is gene 1, it has its own promoter P1. And let's say this is gene 2, and let's say another gene is here, P2, gene 2. So every gene has their own promoter in eukaryotes. One gene, one protein, right? However, if you look at the prokaryotes, they have multiple genes. The gene 1, gene 1, 2, 3, 4, and 5. Under this promoter, we call it P. So multiple genes under the same promoter, and there is also this operator O. So P, O, and these structural genes which makes the protein. And why they do so? Because they're very simple. They don't have room for intron. So every gene is working as it is, like it's to its full capacity. Only difference is that highly economic system in prokaryotes, because these genes like 1, 2, 3, 4, 5, maybe their products, these 5 proteins, they work at one place where they can metabolize, like, their metabolic activity is needed at one place to process that substrate, let's say, and these enzymes are needed in that work together. So all these genes means all these genes are under the control of same promoter, and we call this as a operons. Operons are found only in prokaryotes. First thing you have to know that. It's the most studied part in that bacterial gene regulation in prokaryote because it's simple. It's very, very difficult to study these things in eukaryotic system because of the complexity of genetic system or other factors, okay? So here I made this slide for you guys just to know that inducible operons, because it's like you can switch these lights on. So definitely in the initial mode, they are in off mode and need to be switched on. So we call it as a inducible lights. On the other hand, repressible operons are working normally in the on mode, and you need to switch them off. So for example, if I need the lights, when we don't need the lights in the house, genes, don't need them, lights are off mode. When we need them, we switch them on. So same thing, same analogy, you can use it. So they have taken an example of lac operon. We call it lac operon, means operon that consists of these 3 enzymes, LAC-Z, LAC-Y, LAC-A, and they make these 3 enzymes which can hydrolyze lactose. They bring inside the cell, and this is also does something, but we don't know for sure. These are the names of these enzymes. That is a structural gene. They have operator. They also have a regulatory gene, which can control, the regulation of, or like control the activity of lactose operon. So let's see how it does. In normal scenario, that's how it looks like. Here we have 3 genes. They make lactose A, ZYA, these 3 genes. If transcription is on, these are the 3 products we are going to get. One thing to be noticed here, that this gene is transcribed as a one single mRNA molecule. And each gene has a start and stop here. That's how it's possible to make 3 different proteins. And all the 3 proteins or enzymes, they work to process a lactose. That's why we call it as a lactose operon. There's another one part here, we call it as a regulatory gene. The function of this regulatory gene is to make a protein. We call it as a repressor or inhibitor, right? This repressor, if it's active, it will go and bind here and it can stop this process, okay? In a minute, I will show you that. But right now, what you can see, that this regulatory protein has a promoter which makes this transcript and then translation product, which is shown in red, and it's in inactive form. It cannot bind here. So this process is on and this is also on, and you're getting protein. Now I will go into the detail. Lactose operon, when lactose is not in the media, lactose is not in the medium, the plates you are talking about, we are using, lac operon is in off mode. Initially, off mode. How? That's how they are showing. You have to read it, very important part. Everything is explained here. So regulatory gene makes a repressor. Repressor is active, it binds on the operator. Now promoter, this RNA polymerase cannot bind here. So no transcription, no transcript, no translation, nothing. That's how it keeps the genes off mode. On the other hand, in next slide, we say that this gene, we need to switch it on. So lactose, in the presence of lactose, when you have lactose in the medium, it acts as a inducer. What it does? Induction of transcription. How? It binds to this repressor and make it inactive. Earlier it was active. It could bind here. It was active mode. Now if you add the lactose, it will become inactive and it will not be able to bind here, right? It will come off from here and your RNA polymerase is able to go in this direction, read these three, make the mRNA. Once you have mRNA, it can make the 3 different proteins and they can process lactose. Lactose is a disaccharide. You have to break it down into glucose and galactose, and then cells can utilize those small molecules for making use of energy. On the other hand, arginine is a amino acid which is found in like a more quantity in the cells. It's like a feedback loop inhibition. This operon is in normally on mode. So you can see it's in on mode. RNA polymerase making mRNA, mRNA is making proteins. These enzymes are responsible for making arginine, which is one of the amino acid. Now, when cells have excess of arginine, they need to stop it. So arginine acts as a inhibitor, kind of, it will act as a co-repressor because the repressor is here. Right now it's in inactive form. This will bind to the repressor, and in the next slide you can see repressor is active. It can bind to the operator, and further this RNA polymerase cannot go, and there is no mRNA, no protein. So these are two ways that your operons can be on, it can be off mode. And the last part, they just talk about this new field, bioinformatics, that they use these computer like models to study that where the drug can bind on the 3-dimensional structure of a protein. It's like use of computer, like technology these days with the information we have in bio. So we have the genome of Haemophilus influenzae. Bacterial E. coli has been also sequenced, means we know all the genes present and their sequence. And now we are also having this project where we are trying to find microbiome project where, we can find different bacteria in healthy people in their nose or in their body, and we can find the differences, that why some people are healthy, so why some people are not healthy, what is the role of these good bacteria in our body. So a lot of projects actually have been started, and we can track livid health issues we have, or anything in future will come from those discoveries. So that's chapter 8. And if you have any question, just let me know. I can quickly show you a video that can also show you something from DNA to protein. 1 second.
Here is a cell, the basic unit of all living.
1 second. So you will understand here like the DNA to protein. So I hope there are a lot of other videos you guys can watch, come back to understand,
Tissue. There is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
Okay, so that's the DNA to protein.